This video is on molecular pharmaceutics, specifically nanotechnology and targeted DDS and TDS for MPharm and Pharmaceutics, Semester 2, Unit Number 4, Part 3, presented by Dr. Krishna Yadav. We begin with pressurized metered dose inhalers, or PMDIs. They work in 4 key steps: preparation, inhalation, actuation, and breath hold. Proper coordination is essential for optimal drug delivery to the lungs. PMDs deliver an emitted dose of 20 to 100 micrograms per actuation and have a fine particle fraction of 10 to 40%. Advantages include portability and compactness. Limitations involve coordination, high oropharyngeal deposition, and the cold Freon effect. PMDs come as solution or suspension MDIs. Solution MDIs dissolve the drug in propellant and co-solvents, offering stable, consistent dosing, with examples like beclomethasone dipropionate. Suspension MDIs use micronized drug particles suspended in propellant but face stability challenges like Ostwald ripening, as seen in salbutamol formulations. Dry powder inhalers, DPIs, are propellant-free devices that rely on the patient's inspiratory effort. The mechanism involves loading, preparation, inhalation, deposition, and a 5 to 10 second breath hold. High inspiratory effort is key for efficient drug dispersal and delivery. DPIs are classified based on dose design: unit dose devices, multi-unit dose devices, and multi-dose reservoir devices. They are also categorized by energy source as passive, active, or third-generation DPIs, which incorporate electronic controls. Powder formulation strategies for DPIs aim to achieve adequate powder dispersion. These include carrier-based formulations, like those using lactose, carrier-free formulations using techniques like spray drying, and engineered particles, such as large porous or hollow particles, are also used. Advantages of DPIs include being propellant-free, breath-actuated with no coordination issues, and suitable for high-dose drugs. Limitations involve dependence on inspiratory flow rate, moisture sensitivity, and the need for complex powder engineering. Nebulizers convert liquid drug formulations into aerosol droplets for inhalation. Key types include jet, ultrasonic, vibrating mesh, and soft mist inhalers. Each type has distinct principles, advantages like delivering high doses or being silent, and limitations such as long treatment times or the risk of protein degradation. Comparing pulmonary devices reveals differences in propellant use, coordination required, and inspiratory effort. SMIs and PMDs show excellent portability. Lung deposition varies, with SMI having the highest range. Jet nebulizers are suitable for all ages, but the device cost is generally low. Preparation of MDI suspension formulations involves drug micronization, container preparation, formulation preparation, filling, and quality testing. Solution MDIs are simpler, involving drug dissolution, filling, and stability testing. Carrier-based DPI formulations are prepared through drug micronization, carrier preparation, blending, capsule or blister filling, and device assembly. These steps ensure appropriate particle size, blend uniformity, and sealed dosing. Carrier-free DPI formulations use techniques like spray drying, which produces dried particles, and spray freeze drying, creating porous, light particles. Supercritical fluid technology and thin film freezing are other methods to produce particles for inhalation. Preparation of nebulizer solutions involves formulation development, typically using sterile water or saline, and optimization of pH and osmolality. Sterile manufacturing follows with filtration. Packaging involves unit dose vials or multi-dose bottles, with caution advised for benzalkonium chloride due to bronchoconstriction risk. Aerodynamic particle size distribution, APSD, is critical for evaluating inhalation products. Cascade impactors like the ACI, NGI, and MSLI, along with the twin impinger, are used to characterize aerosol particles, with different flow rates and USP methods applied depending on the device type. Cascade impactors function by trapping particles based on inertia. Larger particles are retained in the first stages, while smaller particles pass on to subsequent stages, eventually passing out of the last stage. Key APSD parameters include fine particle dose and fraction, emitted dose, and mass median aerodynamic diameter. These metrics characterize drug delivery efficiency, particle size distribution, and the proportion of particles capable of reaching the deep lung. The aerodynamic particle size distribution is a golden metric for respiratory deposition. FPD and FPF quantify the mass of drug below 5 micrometers. MMAD indicates the diameter where 50% of aerosol mass is larger and 50% is smaller. Additional in vitro tests include delivered dose uniformity, which checks consistency across actuations. Spray pattern and plume geometry characterize the spray cloud. Leak tests confirm container integrity, and moisture content is critical for DPIs to prevent degradation. Specific tests for DPIs assess flow rate dependence and powder flow properties. Capsule or blister emptying is measured, and specific resistance determines the required inspiratory effort. For nebulizers, nebulization rate and droplet size distribution are measured. In vivo evaluation of inhalation products involves assessing systemic absorption, therapeutic performance, and lung deposition after administration. Pharmacokinetic studies measure drug concentration in blood or plasma over time, and evaluate parameters such as Cmax, Tmax, AUC, and bioavailability. The charcoal block technique is often used to block gastrointestinal absorption, so that only pulmonary absorption is assessed accurately. Pharmacodynamic studies evaluate the clinical response produced by the inhaled drug. For bronchodilators, important parameters include FEV1 and peak expiratory flow rate, or PEFR. These studies help determine dose-response relationships, along with onset and duration of action. Gamma scintigraphy is an advanced imaging technique used to visualize lung deposition. Here, the drug is radiolabeled with technetium-99 meters, and gamma camera imaging shows the regional distribution of aerosol particles within the lungs. It is considered the gold standard for in vivo deposition assessment. Stability testing for inhaled products is conducted according to ICH guidelines, monitoring key parameters like drug assay, degradation products, delivered dose uniformity, and physical appearance over shelf life. These references offer further detailed information on inhalation drug delivery techniques, products, and pharmaceutical inhalation aerosol technology.